Hello everybody. Today we will discuss the different components that is present in the neuron. So let's get started. First of all, we will look at the three most important components of the neuron. First is the cell body and second is the neurites. In the neurites, you can either see the dendrites that carries message towards the cell body, and you can see the axon that carries message away from the cell body. So first organelle, or the first most important component that is present in the cell body, is the nucleus. Nucleus is double enveloped organelle, and most important component in the nucleus is the nucleolus. That is the most important factor that assembles the ribosomes. Inside the nucleus, you will also see the presence of the genetic material like DNA, and along with that, you will also see the bar bodies. Bar bodies present only in case of female, because in female you will see the pair of sex chromosome like XX. In female, there is a sex chromosome XX, and in male there is a X and Y sex chromosome. In female, one X chromosomes becomes inactivated, and now due to the inactivations of the X chromosome, it will look in the microscope as bar bodies. These bar bodies present just beneath the nuclear envelope. So this is important thing. Bar bodies will only be seen in case of female. And what type of female? Mammalian female. So that is the important thing you have to remember about the bar bodies. Okay, next move towards the second organelle, that is the Golgi complex. Golgi complex basically modifies the proteins made from the ribosomes. So if I say protein attached with the carbohydrate, then that will become the glycoprotein. If the lipid combined with the protein, then it will become the lipoprotein. So modification happens in the Golgi complex organelle. Next is the centrioles. Centrioles are the tube-like structures present in the cell body. Centrioles having two functions. In the non-dividing state, it helps in the formations of the microtubules, that is the component of our cytoskeleton. And in the dividing state, centrioles makes spindle fibers. So in spindle fibers happens in the immature neurons, because mature neurons do not divide. Next is the mitochondria, important component of the energy production inside the cell. And next is the Nissel's granules, most important. These are the granules of the rough endoplasmic reticulum. Endoplasmic reticulum having ribosomes on it. So these Nissel's granules most important, and they spread all through the cell body and at the proximal parts of the dendrites also. These can also be present at the proximal parts of the dendrites. But these Nissel's granule will not be present in the axon hillock. At the periphery or proximal side of the axon, you will see the axon hillock, and this is the location where Nissel's granules do not present. So that is the important thing. And other important thing, at some stages, Nissel's granules disappear. Basically, these granules starts accumulating at the periphery of the cell body, and ultimately these granules will not look in the center of the cell body. So that state is called the chromatolysis. Next is the microfilaments. Microfilaments made up of actin protein. These filaments present just beneath the cell membrane of the cell body, and these filaments help in the movement or transportation of the different organelle, and it also help in the formations of the neurites in the cell bodies. Okay, and along with that, you will also see the presence of the neurofibrils. Neurofibrils made up of neurofilaments. Bundles of neurofilaments combine, and it will result in the formations of the neurofibrils. That is the important component of the cytoskeleton and maintains the shape of the cell bodies. And along with that, you will also see the microtubules. Microtubules, these made up of tubulin protein, and this is the thick cytoskeleton component of the cell body. Okay, next you will see the summary of the different components present in the neuron. First is the nucleus. In the nucleus, you will see the nucleolus, that is the important component of the ribosome assembly, and bar bodies present only in case of female. And what is bar body? Bar bodies are actually inactive X chromosomes. Next is the Golgi apparatus that makes modification like glycoproteins and the lipoproteins. Next is the mitochondria, important component of energy production. Lysosomes. Okay, we have forget this. Lysosomes having hydrolytic enzymes that break down the food. So it has three types: primary, secondary, and the residual body. Primary, newly formed lysosomes are the primary lysosome. When the lysosomes attach with the food particle, then it will become the secondary, and residual body, in which you will see the inactive enzymes in the lysosome. Next is the rough endoplasmic reticulum, a reticulum having ribosomes attached on it, and it ultimately results in the synthesis of the proteins. Smooth endoplasmic reticulum also present in the cytoplasm. Nissels granules, these are the accumulations or the granules of the rough endoplasmic reticulum. So ultimately, rough means it also involves in the synthesis of the protein. Cytoskeleton, you will see the neurofibrils made up of neurofilaments, and the size diameter of the neurofilaments is 10 nanometer. Next is the microfilaments having diameter 3 to 5 nanometer, and microtubules 25 nanometers. Next is the centrioles involved in the spindle fiber and involved in the maintenance of the microtubules. So these are the important components of the neuron. Next important thing, neurofibrils made up of neurofilaments. These are present throughout the cell bodies. That is the important thing. And microtubules made up of tubulin proteins having large diameter as compared to the other filaments. Basically, movement of the organelles will be done with the help of the microtubules. So movement can be two forms: towards the cell body and away from the cell body. So movement can be towards the cell body and away from the cell bodies. So that is the important thing. So movement can be rapid and the slow. Rapid movement, it will take the 100 to 4 or 100 millimeter per day. This is a small distance covers in one day, 100 to 400. Movement can be antegrade and the retrograde. Antegrades mean away from the cell body, movement away from the cell body. And in that case, you will see the organelles coated with the kinesin protein moves the organelle through the microtubules. If I say here is the microtubules and here is the organelle having kinesin protein on it. Now this organelle, kinesin coated organelle, move alongside the microtubule. So that is the movement, antegrade movement, and in that case you will see the movement will be away from the cell body. In the retrograde, movement will be towards the cell body, and in that case you will see the dynein coated organelles will be helpful in that case. So rapid movement can be antegrade and the retrograde. And in the slow, you will see the 0.1 to 0.3 millimeter per day. This is a small distance covered in one day. But in that case, cytoplasmic flow helpful in that case. And in that case, you will see the only antegrade movement will be seen. So this is all about the different components present in the neuron. But if you still have any question, you may ask in the comment section. Thank you so much.